Always a tricky topic, meiosis. So let's have a look through. So I've made a few diagrams of some cells. So I'm just going to kind of make some observations about these. This is a cell, it's got 4 chromosomes, it's got 2 pairs, okay? And because its chromosomes are in pairs, it's a diploid cell. So first thing I'm going to write here is that, yeah, this is a diploid cell. And we write that as 2n, okay? And it's, yeah, so it's diploid, and I can also say that its diploid number is, well, it's 4. It's the number of chromosomes. So the diploid number for this cell is 4. So if this was a human cell, like a cheek cell or a stem cell in the testes that is going to make sperm by meiosis, it'd be 2n, but the diploid number would be 46. Okay, we have 23 pairs of chromosomes. The diploid number is 46. Okay, here I have a pair of chromosomes. So what kind of pair of chromosomes are these? Well, they're the same size, and if I looked at them genetically, they carry the same genes. So these are an homologous pair. And as we know, a homologous pair means they carry the same genes at the same loci. They differ by their alleles. Okay, and there's another pair there that's smaller. The other observation I can make is that, well, they consist of 2 chromatids, so each chromosome has been replicated. So it's pairs of chromosomes, and they've been through replication, been through semi-conservative replication. Again, yes, so chromatids often gets confused with chromosomes. So I'll just get my pointer, let's go back to this diagram. Oops, scroll down a bit. So this red one here, this is our chromosome. It's gone through replication, therefore it's made of identical chromatids. Okay, so there might be a gene at this location, it will be the same gene on this location. Same gene on this location, same gene on this location. However, this might be a dominant allele of a gene, so you have brown eyes, have brown eyes. This might be a recessive allele of that same gene, and therefore say have blue eyes, have blue eyes. Okay, so yeah, chromosome, homologous pair of chromosomes, and then a chromosome made of identical chromatids. Okay, right, so that's our first diagram. Let's see how we get on with the next bit. I'm not going to go through all the detail of meiosis, because you have a textbook. If you want to see a whole story, then you can, there's numerous animations that show it far nicer. Remember the McGraw-Hill ones? If you're ever stuck for, I don't understand something, just type into YouTube McGraw-Hill and then follow it by another word like meiosis. And there'll be 1 or 2 animations narrated by a guy normally with a very American accent. They'll go through it, or these other links that I put on the other email I sent you last week. So what's going on here? Well, here this is a stage of meiosis. We don't need to know any stages of meiosis. I don't need to know the difference between metaphase 1 and metaphase 2. This is metaphase 1 because the chromosomes have aligned either side of the equator of the cell, okay, either side of the metaphase plate. And in mitosis, they line as a single line. Here, they line across the equator, okay, and that positioning is random. Okay, so first of all, we're just going to say what's happened here. So how can I explain this bit here? What's happened there? Well, that's called, as you're all shouting at me, yeah, crossing over. And that resulted in, well, they've swapped the same length of DNA. So it's resulted in, really, the mixing of alleles. Between these two chromosomes, and it's the, between maternal, inherited from mother originally, and paternal chromosomes. Chromosome in this case. And in crossing over, that's when, again, you just need a two-mark definition of it. It's when the same length of DNA is exchanged. In year 13, we do another type of mutation where not the same length of DNA is exchanged, and that leads to duplication mutations where you double up a gene on a chromosome. But again, we can worry about that in year 13. 2021, that's when we have to worry about coming back to meiosis to look at other mutations. And it also means that you're not going to get a proper, your final exam is at the end of, at the end of year 13. So we are going to meet meiosis again slightly. There's going to be time in school to pop along to a workshop anyway, but because it is a notoriously tricky topic. Okay, so that's crossing over. Remember, the same length of DNA is exchanged between homologous chromosomes. Therefore, we're mixing alleles between maternal and paternal chromosomes. I'm just going to add on homologous pairs, on homologous. In fact, it's pretty the same between, isn't it? Between homologous chromosomes. Because they can do it in error as well. Also, I'll put the mainly red one on this side, on the left side, and the mainly blue one on the right side. And they have to pair up. But I've done them the opposite way around for the smaller chromosome. And these haven't crossed over. Crossing over is random. It may or may not happen. Where it happens is also random. So I could have put even a smaller blue bit on here or a bigger blue bit. They can cross over here and at the tops as well, or not at all. So yeah, here I've got red, blue, and here I've got blue, red. So the position either side of the equator is what we call independent segregation. So the position of these is random. Equals independent segregation. Okay, so yeah, we are mixing up maternal and paternal chromosomes. Okay, so this cell is going to then split. Yeah, you're going to get a cleavage furrow forming here, and then that's going to form into two cells that are then haploid. And then they have to finish off their division by splitting again so that we end up with not a chromosome made of chromatids. I need to separate this side from this side. It's annoying that my pointer is red and the chromosome's red. Okay, so it's going to go through two sets of cell division. One now to produce haploid cells with one of each chromosome pair, and then it does it again to get back to single chromosomes, not those made of chromatids. Right, so I'm on my last diagram now. So this is now the result, and you can see that I've got 4 different cells produced. 2 of these are like normal cells. So this one here, so this is a haploid gamete. It's now N. It's got 1 of each pair. And so is this as well. Yeah, those are normal haploid gametes. They'll like grow a tail if they're going to be a sperm cell, and off they'll go to do their merry business. If you look here, this is the one that's got a bit of maternal chromosome with a bit of paternal chromosome. So we've mixed up the alleles of genes. So this cell here is mainly red with a bit of blue. This one's is entirely red, so these have got different alleles of genes, and these have got copies of the same chromosome, so there's no difference between this one and this one, but in the entire cell, then we have got differences. Okay, then I've got these two cells over here, and this is where a mutation has occurred because there's a change in the amount of DNA in a cell. Okay, so this just means that this chromosome here, it failed to separate. Instead of spindle fibers attaching on both sides of it, they've attached on one side only and dragged it into one cell and not separated the chromatids. So this one here, the chromatids fail to separate. So it's called a mutation. So we call this a mutation because it's a change in the amount of DNA. So yeah, it's a change in the amount of DNA in a cell. So if this cell becomes a sperm cell, it's missing another chromosome, and therefore when it fertilizes an egg cell, there'll be a missing chromosome, and that generally leads to the cell not dividing any further. There's a significant problem. Sometimes, though, it is allowed to kind of take place, especially if it's a sex chromosome in humans, because they're less kind of crucial for development. So yeah, you can get syndromes called Turner syndrome, where there's a female who's only got one X chromosome in her cells. She's got all the other 22 pairs of them, but there's only one X chromosome, and normally there's a pair of X chromosomes, and therefore that leads to certain health effects, and it's called Turner syndrome. Likewise, you can get a male who's got XXY. So maybe there's two copies of the X chromosome somehow fertilized by a sperm, therefore you get XXY, therefore a Y chromosome causes maleness, and but there's a host of issues because of it. Normally sterility, because there's an uneven number of chromosomes. So this doesn't stay stuck together by its centromeres, that will divide over. Just show you what that will do. So this will disappear. Wait, it won't disappear. It'll stay there, but then it will just, it ends up separating anyway. So you've just got two copies of it, and you see how that will cause chaos in a cell. So again, in biology, in the exam question, asks you to apply your knowledge of meiosis to different scenarios, and it might be, describe how a zygote, a fertilized egg cell, is produced for someone who's got Klinefelter syndrome, who's got XXY chromosomes. So therefore, the answer would be, well, it would be due to a non-disjunction mutation, where in meiosis, the chromatids fail to separate. Therefore, the gamete that was produced wasn't haploid fully. It had a pair of X chromosomes, and therefore when fertilized, a Y chromosome carrying sperm came along, and therefore ended up with an XXY offspring. And that's really it. Again, it is tricky. You need to, if nothing else, make sure you can do your definitions. What's crossing over in 2 marks? What's independent segregation in 2 marks? And what's a non-disjunction mutation in 2 marks? What's a haploid cell? What's a diploid cell? Oh, the last thing that I need to say is, what's the haploid number of this cell now? Well, this cell's got a haploid number of 2. Yeah, that's its haploid number. It's just the number of chromosomes. And we started off with a cell that had, this is where I always mess up when I'm trying to scroll around here. Remember, we started off with a diploid number of 4. There it was. So yeah, I shall stop there. because it's 15 minutes long enough.